The focus of this lecture will be to examine the vast diversity of prokaryotic organisms. Prokaryotic organisms are immensely diverse, totaling at least 5,000 known species. Some prokaryotes, such as archaebacteria, can thrive in habitats that are way too extreme for eukaryotes and are thus otherwise known as extremophiles. Prokaryotes can also perform metabolic activities upon which all other life forms on Earth depend because of their crucial role in recycling nutrients throughout ecosystems. This diagram represents a phylogenetic tree, which organizes life forms according to their respective evolutionary relationships. In general, the closer two groups lie on the tree, the more closely related they are. For example, archaebacteria such as methanogens and extreme halophiles are more closely related to one another because they share a more recent common ancestor, represented by this branch point, than either group does to the hypothermophiles. Not surprisingly, this phylogenetic tree shows that the evolutionary lines of prokaryotes such as the bacteria and the archaea, as well as all eukaryotes, can be traced back to a single common ancestor that existed somewhere around 3.9 billion years ago. From this universal ancestor, it was the bacteria that were the first to evolve. From previous discussions, we should recall that these prokaryotes were anaerobic heterotrophs. From the bacteria evolved archaebacteria, which can tolerate and thrive in environments of extreme acidity, salinity, and temperature, and are thus also referred to as extremophiles. Finally, from certain species of archaebacteria evolved the eukaryotes. Therefore, these two groups are more closely related to one another because they share a more recent common ancestor than either group is to the bacteria. Structural and genetic evidence supporting this close evolutionary relationship between the archaebacteria and the eukarya will be further examined towards the end of this lecture. With regards to the structure of prokaryotic cells, one of the main areas of focus will be the cell wall. Whereas the cell walls of archaebacteria are composed primarily of cellulose, those of bacteria are comprised of a polysaccharide known as peptidoglycan. The two subunits that comprise the peptidoglycan of the bacterial cell wall are N-acetylglucosamine, also referred to as NAG, and M-acetylmuramic acid, also referred to as NAM. These subunits are also referred to as amino sugars because while bearing resemblance to a typical monosaccharide, they also bear a modified amino group. As shown here, the NAG and NAM subunits that comprise the peptidoglycan polymer are arranged in an alternating fashion. Additionally, each peptidoglycan chain is cross-linked or joined to one another by short peptide cross-bridges. As we will see shortly, it is the amount of peptidoglycan as well as the complexity of the bacterial cell wall that serves as a basis for classifying all bacteria as either being gram-positive or gram-negative. Gram-positive bacteria exhibit a relatively simple cell wall structure with an abundance of peptidoglycan which lies exterior to the plasma membrane. When a gram stain is applied to such bacteria, the colonies, as you can see, take on a purple appearance. This technique, which we will apply in lab, allows us to generally classify bacteria based on the complexity and composition of their cell walls. Once a bacteria has been identified as either gram-positive or gram-negative, more specific techniques can be applied to identify the bacterial species. In contrast to gram-positive bacteria, gram-negative bacteria exhibit a much more complex cell wall structure that incorporates less peptidoglycan. As in gram-positive cells, the cell wall of gram-negative species lies exterior to the plasma membrane. As this diagram illustrates, the cell wall of a gram-negative cell consists of a single layer of peptidoglycan, which lies directly below an outer membrane which incorporates a series of lipopolysaccharides. These molecules can be released from the cell upon its death and act as endotoxins. We'll discuss more about the activity of endotoxins at the end of this lecture. As we can see, when a gram stain is applied to such cells, the colonies take on a pink appearance. In addition to the cell wall, we can also examine prokaryotic structures such as the flagella, which is composed of a filament, a hook, and a basal apparatus. With regards to the filament of the prokaryotic flagella, it is a rigid, hollow, helical structure made out of the protein flagellin. In contrast, the flagella of eukaryotic cells is made out of the protein tubulin. The basal apparatus of the prokaryotic flagellum serves to anchor the structure into the cell wall. In addition, it acts as a motor to rapidly rotate the filament. Thus, the prokaryotic flagellum adopts a rotary motion. In contrast, the eukaryotic flagellum bends back and forth. The hook component of the flagellum simply serves to couple or link the filament to the basal apparatus. With regards to the genome of prokaryotic cells, the DNA is relegated to a region of space called the nucleoid. In bacteria, this DNA, which is organized into a single circular chromosome, lacks non-coding regions called introns. As we will later see, these non-coding regions are evident in archaebacteria and eukaryotes, once again suggesting a close common ancestry. The circular chromosome of prokaryotes can give rise to small, self-replicating entities known as plasmids. Whereas the main circular chromosome contains housekeeping genes which are responsible for regulating the cell's metabolism, the plasmid, by contrast, contains very little genetic material. Although plasmids are not essential for maintaining a cell's metabolism, they can contain genes that can confer upon a cell resistance to antibiotics, and thus provide the cell a survival advantage in the presence of these compounds. As previously discussed in class, the prokaryotic ribosome is designated 70S compared to the eukaryotic ribosome, which is designated 80S. These designations are a reflection in differences in the makeup of each structure and their respective densities. The differences between the prokaryotic and eukaryotic ribosomes is evidenced in the prokaryotic ribosome's susceptibility to certain antibiotics such as tetracycline, to which the eukaryotic ribosome is unaffected. The mechanism by which prokaryotic cells divide is known as binary fission. Binary fission is an evolutionary precursor or forerunner to mitosis exhibited by eukaryotic cells. One major difference between binary fission and mitosis is the lack of a spindle apparatus which would otherwise evenly separate the genetic material. In the absence of this structure, the genetic material of the cell is separated by attaching to the cell membrane. As the cell elongates, each copy of the genome is separated, ultimately leading to the division of the cytoplasm and the establishment of two genetically identical cells. As a result of cell division, bacterial populations tend to initially exhibit exponential growth, in which the population size doubles per unit time. As a result, the growth rate, as seen here, is relatively constant with a positive slope. Exponential growth patterns are generally observed when resources within the environment are unlimited, such as they would be in a test tube. In reality, exponential growth cannot be maintained indefinitely because the population size will begin to plateau as resources become increasingly more diminished, and thus the growth rate will exhibit more of an S-shaped curve representative of logistic growth. The term carrying capacity refers to the maximum number of individuals that the environment can support based on the number of available resources. An inverse relationship exists between carrying capacity and resource availability within an ecosystem. In other words, at times when resources are abundant, the carrying capacity increases, resulting in a corresponding increase in population size. Conversely, at times when resources are less abundant, the carrying capacity will decrease, resulting in a corresponding decrease in population size. As previously discussed, all prokaryotes replicate asexually by means of binary fission. Although we would expect individuals to be genetically identical, what we actually observe is that there is a great deal of genetic diversity within bacterial colonies due to three mechanisms that function in genetic recombination. These mechanisms include conjugation, transformation, and transduction. These recombination mechanisms are vital for establishing and maintaining sufficient genetic diversity which would allow colonies to adapt to changing environmental conditions. The first of these mechanisms, known as conjugation, involves the pairing of cells by way of a cytoplasmic bridge known as a pilus. It is across this bridge that DNA can be exchanged between the cells, leading to genetically recombined or genetically unique individuals. The second mechanism, known as transformation, involves the uptake of foreign DNA by living cells from cells that have died and have broken down. As a result of absorbing this foreign DNA, the living cell is able to express potentially adaptive traits that may allow them to better survive in their surrounding environment. The last mechanism, known as transduction, involves the transfer of genes between bacterial cells by way of viral particles. As seen here, upon landing on a host cell, a viral particle will inject its DNA, upon which the host will produce new viral particles. As shown here, these new viral particles are packaged with a copy of the virus's genome. In some cases, however, shown here, the viral particle may be packaged with part of the bacterial host's genome. Upon infecting another host, this viral particle can then introduce that foreign DNA into the host cell, thereby transforming it, enabling it to express novel traits that may be adaptive to new environmental conditions. It's important for us to realize that without these three methods of genetic recombination, all cells within a bacterial colony would be genetically identical. Consequently, in times of environmental change, it would be very unlikely that any one cell within a colony would have traits enabling them to tolerate these changes and survive to reproduce. Prokaryotes generally cause disease by producing various proteins known as exotoxins and endotoxins. With regards to exotoxins, they are generally observed to be produced by both gram-positive and gram-negative bacteria. These exotoxins are proteins that are secreted from these cells and generally travel far from the site of infection and target body systems specifically. As we can see, they are generally highly toxic and in many cases often fatal. An example of an especially potent exotoxin would be the tetanus toxin produced by the bacteria Clostridium tetani. This particular toxin targets muscle tissue, preventing muscles from relaxing. As a result, the victim experiences spastic paralysis. Some of the symptoms associated with this condition might be lockjaw and asphyxiation leading to death if the respiratory muscles are infected. In contrast to exotoxins, bacterial endotoxins are produced primarily by gram-negative bacteria and consist of the lipopolysaccharides within the gram-negative cell wall. These compounds are generally released when the cell dies and breaks down. Unlike exotoxins, which specifically target various tissues throughout the body, endotoxins have more of a general effect on body systems, causing fever, diarrhea, so on and so forth. As a result, these particular compounds are very seldom life-threatening. The following table summarizes many of the characteristics exhibited by members of the bacteria, the archaea, and eukarya. It's extremely important that you're able to identify and elaborate on some of the key differences between these three groups. And in studying this table, you should recognize that the archaea and the eukarya tend to share more characteristics in common with one another, which suggests a close common ancestry.